Good afternoon, everyone, and welcome to our Thursday afternoon educational webinar. I'm Melinda Bikini, the Director of Advocacy with the Cholangiocarcinoma Foundation, and today I am thrilled that we will be joined by Dr. Valerie Jansen, who is the Chief Medical Officer at Elevation Oncology. She's a physician scientist with more than 10 years of experience in medicine and oncology drug development. And prior to transitioning to the pharmaceutical industry, Dr. Jansen began her career in academia, focused on understanding mechanisms of a resistance to cancer-targeted therapies. She also cared for patients with cancer as a board-certified medical oncologist. And at Elevation Oncology, she directs a team of medical and scientific professionals who are passionate about developing the right medicine for the right patient. Dr. Jansen will be doing a presentation. Feel free to put questions in the chat or the Q&A box as she's going through the presentation. And then after the presentation, we will have a question and answer session. And I will turn it over to you, Dr. Jansen. Thank you for being here.
Great, thank you, Melinda. And first of all, I would like to thank the Cholangiocarcinoma Foundation for this great opportunity for us to be able to hear today to share with you about Elevation Oncology and the first program that we have in place to help patients with tumors that harbor NRG1 fusions. And I also want to thank all of you who are joining us and your loved ones and caregivers. And I hope that you'll be able to take something away from this, this webinar and help patients just like yourselves and others. So I'll begin by sharing with you the vision of Elevation Oncology. Our vision is that we want to elevate precision medicine to the forefront of every cancer patient's treatment journey. We believe that every patient should know what's driving their cancer and to be able to have access to medicines that treat that specific cancer. We know that each patient is unique, and just like unique patients, tumors are also very unique. And so as Melinda mentioned, prior to me joining Elevation and in the industry, pharmaceutical industry, I was a practicing oncologist and taking care of patients. And nothing would frustrate me more than seeing a patient who have seen multiple lines of therapy but have not yet received genomic profiling. Or likewise, having a genomic profiling of that tumor, but not be able to have access to medicines that could potentially help that patient. And so we at Elevation really want to change the way that medicines are developed for patients living with cancer. Now I want to start with this slide, and it highlights the different genomic alterations. As we know, cancer is driven by different alterations that occur that lead to uncontrolled growth and proliferation. So we know that genes can be overexpressed or amplified. Genes such as HER2, you may have heard of HER2 positive gastric cancer or HER2 positive breast cancer. We also know that mutations can occur in gene that leads to uncontrolled cell growth and proliferation. Genes like EGFR mutations in non-small cell lung cancer. And finally, there are rare genomic alterations where there is a fusing of two genes, and these gene fusions can lead to uncontrolled growth and proliferation. Now, the prevalence of these different alterations also make it more a driver versus a passenger in terms of whether that is the one and only thing that's driving that cancer. As you can see here from bottom to top, gene amplification and overexpressions are common. In terms of they're readily found in terms of in tumors, but they're most likely going to be passenger or a bystander and not necessarily the one and only thing driving that cancer. Contrast this with gene fusion, just like FGFR gene fusion in cholangiocarcinoma or NRG1 gene fusion in a variety of solid tumors. These are low in prevalence. They're rare, but if you find them, they're probably the thing and only thing that's driving that cancer. And therefore, if we can make a medicine that targets that, we have a high probability of success in terms of stopping that cancer growth. And that's what Elevation is focused on, to really be able to help patients regardless of how small or how rare that genomic alteration is. Now, to go into a little bit more about NRG1 fusions, what are NRG1 fusions? So NRG1 gene fusions is the fusion of NRG1 with another gene. And then this results in an abnormal NRG1 fusion protein. These fusion proteins, as depicted in the orange and blue dot, can now activate HER3 or a number of ERB family, but particularly HER3. Now, it stands to reason that if there's a HER3, there must also be a HER2 and a HER1. And in fact, there are 4 ERB family members. So there's HER1, HER2, HER3, and HER4. So these NRG1 fusion proteins activate HER3. When HER3 is activated, it can dimerize or partner with its other ERB family members, particularly HER2. And this results in a signal to the cancer cell to grow and divide. Now, there are medicines out there that target the kinase function of these ERB family. You may have heard of afatinib. This is a kinase, a medicine that targets the kinase function. However, HER3 is unique in that it does not have a kinase function. So you can imagine that afatinib may not be that most ideal medicine to target NRG1 gene fusions. So we have developed a medicine called Cerebentoma. It's a monoclonal antibody that I'll talk a little bit more about in a couple later slides. And we believe that if you can block HER3 with Cerebentoma, then you could potentially shut down that signal to the cancer cell to grow and divide. Now, how do we identify NRG1 fusions in tumors? Well, there are next generation sequencing or comprehensive genomic profiling through a variety of NGS companies. For example, your oncologist can order a test through Tempus or Caris, a Cheyenne, that would be able to identify NRG1 fusions. NRG1 fusions, just like other gene fusions, are best identified through an RNA-based NGS assay. And these assays, these laboratories use that RNA-based assay. Now, it can also be identified through DNA-based NGS, such as that used by Foundation Medicine, but it will have the potential to miss the majority of NRG1 fusions and other fusions as well. As I mentioned, NRG1 fusions, if they're found, they're driving that cancer. And if it is found, it seems to be the one and only thing that's driving that cancer. So we can shut that down, we could potentially stop the growth of cancer. Well, we have also found that NRG1 fusions is identified in a variety of different tumor types. In fact, over 10 different tumor types have been described as potentially harboring NRG1 fusions. Now, NRG1 fusion, as I mentioned, is ultra rare. It occurs in about 0.2% of all solid tumors. Now, it is enriched or more prevalent in certain subtypes of tumors, such as lung cancer, and particularly a type of lung cancer called invasive mucinous adenocarcinoma, or IMA. We also know that in patients with pancreatic cancer, especially those tumors that are KRAS wild type, NRG1 fusions seem to be more prevalent in about 6%. Now, I do call out the biliary tract cancers, such as cholangiocarcinoma and gallbladder. NRG1 fusions have been found in these tumors in about 1% of all of these tumors. So why do we care about NRG1 fusions? Well, we know that it's the thing that's driving that cancer potentially. And unfortunately, tumors that harbor NRG1 fusions don't do as well on standard of care treatment that we have. For example, the standard of care chemotherapy, even combination chemotherapy with 4 or 5 different regimens. Now, this is a study from lung cancer, of patients with lung cancer, and you can see that depending on whether their cancer harbored an NRG1 fusion or not, you can see, unfortunately, that the overall survival is decreased in patients whose tumor have an NRG1 fusion. And this is even regardless of whether you have stage 4 or metastatic disease or stage 1 early disease cancer. So it really signifies that NRG1 fusions confer a worse prognosis. And so we need to do better in terms of making medicines, developing medicines that particularly target this. Now, just to hit home the point a little bit more, this was a study in a global registry of 110 patients, so over 100 patients with NRG1 gene fusion positive lung cancer. Again, you can see in terms of the standard of care that's available for lung cancer, this includes chemotherapy, chemotherapy with immunotherapy, or immunotherapy alone. Now, you also heard me mention afatinib, which is a tyrosine kinase inhibitor. And you can see that even with patients given this medicine, it's only about a 13 to 25% response rate. And unfortunately, a median PFS of anywhere from 2.8 to 5.8 months. So we have to do better. Now, this is in lung cancer, but my understanding is that the Cholangiocarcinoma Foundation is working with patients and caregivers to really define this in also patients with cholangiocarcinoma, so that we can do better in terms of understanding how NRG1 fusions affect cholangiocarcinoma, and then if we can develop medicines that can target that. Now, let me tell you about Cerebentoma, which is the drug that Elevation Oncology is investigating in clinical trial. It is a fully human IgG2 monoclonal antibody. So really what that means is that we have to give it through an IV to patients, and we give Cerebentoma once every week. And the way that Cerebentoma works is that it blocks that NRG1 fusion protein from binding to HER3. Remember, I said that HER3 needed to be activated with NRG1 to be able to dimerize with its partner, HER2, and then sends a signal to the cancer cell to grow and divide. So Cerebentoma blocks NRG1 from activating HER3, and therefore HER3 can't bind with HER2, and therefore the cancer cell doesn't get that signal to grow and divide. Now, Elevation Oncology is a young company. We acquired Cerebentoma from another pharmaceutical company who had tested this agent in over 800 patients. So we know it's safe to give to patients. When we brought this in, we also wanted to optimize the dose for patients with tumors that harbor NRG1 fusion. And so we have determined that giving patients Cerebentoma 3 grams once every week is a good regimen and a tolerable regimen for patients. Now, in the next couple slides, I'm going to show you some mouse models where mice had cancer that harbor NRG1 fusion, and then the mice were treated with Cerebentoma. And you can see how we can show that Cerebentoma blocks the growth of cancer and even shrinks cancer in mice. So this first model is a lung cancer that harbor an SLC3A2 NRG1 fusion. And so on the top graph, you can see that mice were either treated with vehicle or placebo in this, as well as afatinib. Now, afatinib is approved for lung cancer, and the dose of afatinib that's given to humans is 40 milligrams per day. And that is equivalent to a dose of 5 milligrams per kilogram in mice. And you can see that we can increase the dose in mice up to 15 milligrams per kilogram, but in humans, that 40 milligram dose is already pretty toxic, and most patients require a dose reduction. You can see here in terms of Cerebentoma, we have looked at various doses starting at 0.6 milligrams twice a week up to 1 milligram twice a week. And you can see that when we give mice that harbor a tumor that carries the NRG1 fusion, you can see that Cerebentoma is able to block that tumor from growing compared to the vehicle placebo, compared to the afatinib, at least the clinically relevant dose of afatinib. And you can also see that some mice even have tumor shrink such that they're responding to therapy, and some mice even have tumors shrinking more than 50%. Now, this is a second model. This is an ovarian cancer model that has a CLU NRG1 fusion. And you can see here, we also treated the mice with a placebo in green, afatinib 5 milligrams per kilogram, which is the clinically relevant dose, equivalent to the clinically relevant dose in humans in the gray. And in here, you can see we have evaluated Cerebentoma in mice in doses that are going to be equivalent to what we give to patients in the clinic. And you can see here anywhere from 1 milligram twice a week up to 10 milligrams twice a week. You can see when we treat these mice with the CLU NRG1 fusion, these tumors pretty much melt away. And you can see that even if we stop treatment, we can control the growth of the cancer for quite a while. And down below, you can see that all the mice, compared to mice that got afatinib or a placebo, all the mice that received Cerebentoma at all doses had complete or near complete regression of the tumors. So this is quite amazing and really highlights the potential of Cerebentoma then in patients. So what about in patients? So here are a couple examples of other anti-HER3 antibody and used in patients who harbor tumor, who have NRG1 fusion positive tumors. So on the far right-hand side is a HER3 antibody from another company. It's no longer in development, but this was really the proof in terms of blocking HER3 in tumors that are driven by NRG1 gene fusion. This was from a phase 1 study, so finding the dose. And you can see that there was one patient who responded. And when you look back at that patient, their tumor actually harbored an NRG1 fusion. So it stands to reason from this initial patient that if we can block HER3 with a monoclonal antibody approach, then we can stop the cancer growth and proliferation. Now, in the middle is another medicine called zenocutuzumab, which is also in clinical trial by another company. And they show data in patients with pancreatic cancer and lung cancer that indeed by blocking HER3, you can shut down the growth and proliferation of cancer cells that are being driven by NRG1 fusions. This really highlights the potential then for Cerebentoma, Elevation's anti-HER3 monoclonal antibody. And I want to share that example with you in this case study. So this is a patient who was treated with Cerebentoma, and her story was highlighted recently at a meeting by her treating oncologist. This was also an example of how matching the patients with the right medicine at the right time. So this is a young patient, unfortunately, with metastatic KRAS wild-type pancreatic cancer. When the time that she presented with her cancer, diagnosed with her cancer, that cancer had already spread to her liver. And she was started treatment with the standard of care, which is FOLFIRINOX, a 4-drug combination chemotherapy. Unfortunately, she didn't tolerate that chemotherapy very well, and her oncologist then gave her FOLFIRI and dropping one of the drugs that was causing her some trouble. Unfortunately, her tumor did grow through that, and she was treated on another chemotherapy regimen, this time a 2-drug combination with gemcitabine and Abraxane. Again, her cancer grew and really left with nothing else but to recycle her chemotherapy. The patient was put on FOLFIRI again, and her tumor was also sent in for genomic profiling. And fortunately, it did reveal an NRG1 fusion. And we were able to provide her with Cerebentoma under compassionate use because the clinical trial was not yet open in the country that she resides. So here you can see an example of how her CA19-9 was markedly elevated over 1,000. And as you may be aware, a normal is less than 40. And so with her CA19-9 being elevated over 1,000, starting treatment on Cerebentoma alone, you can see this decline. Likewise, on the orange line is each dot represents a time frame that when she went in for her CT scan. And each time that she was scanned, you can see that her tumors were shrinking, such that she did receive what we call a partial response per RECIST criteria. And of course, a picture's worth 1,000 words. So on the left-hand side, you can see here, this is a representative CT scan of one of the liver lesions that she had. You can see here before starting treatment with Cerebentoma, and then after treatment with Cerebentoma, that lesion, that metastatic disease is pretty much gone and can't be seen on CT scan. So it's quite remarkable that, Cerebentoma alone as a monotherapy in this patient's case was able to give her over 9 months of disease control, and then her tumors shrank more than 50% from the beginning before starting treatment on Cerebentoma. And this leads us to our Creststone study that we are evaluating Cerebentoma in adult patients with any solid tumor that has been found to have NRG1 gene fusion. Patients also have been treated with at least one prior systemic therapy, and then patients are being assigned to one of 3 cohorts. Patients are receiving Cerebentoma, and as I mentioned, it's an IV infusion. It's about a 1-hour IV infusion once every week. And our cohort 1 is the cohort that we believe will show the potential of Cerebentoma in patients who harbor NRG1 fusion. So these patients would not have seen prior ERB-directed therapy, so prior afatinib or prior zenocutuzumab or any of the other anti-HER3 therapies. Patients then would also provide tissue for us to be able to look at the NRG1 fusion status and confirm that. Again, we will be looking to see how well cerebrentumab is able to shrink tumors. And of course, we want to understand the continued safety of cerebrentumab in patients with NRG1 fusion positive tumors. Now, we know it's important for patients. What about those patients that have seen prior afatinib? We want to make sure that we explore that as well and to see if cerebrentumab could overcome that. And so those patients are treated in exploratory cohort 2. Likewise, I mentioned that when NRG1 fusion is found, it's typically the one and only thing that's driving that cancer. But we know there might be tumors out there that might have NRG1 fusion with another molecular alteration, like an ALK fusion. And so we want to make sure that we are able to investigate those tumors as well. And so those tumors are investigated in cohort 3. But all patients would receive cerebrentumab at 3 grams once every week. Now, our study is open globally. We are across the US. You can see, and this is not a comprehensive list. It needs to be updated, but you can see we span across the US. We have now also gone into Canada and Australia and looking to expand into other countries because we really want to be able to help patients wherever they are and be able to meet them and their caregivers whenever they need cerebrentumab if their tumors harbor an NRG1 fusion. We also partner with a number of collaborators. These are collaborators that test for NRG1 fusions, as well as collaborators that treat patients. So the US Oncology Network, where it's the largest oncology practice in the US, as well as you may have heard of Caris and Tempest, where we might not have a clinical trial site open, say if you're in Wisconsin, but we have, through Tempest and Caris, the ability to open a clinical trial site closer to the patient's home, and we can do that in as little as 14 days. So really, our mission is to be able to help patients, even with the rarest genomic alteration. And really, Creststone is the beginning. And again, as I mentioned, our vision is really to advocate for patients to know what's driving their cancer and to be able to have access to medicines that target that cancer. And so you can see here, really our goal is to be matching the right patient to the right therapy. And if that's our first track is really with NRG1 fusion and cerebrentumab. We advocate for every patient to know what's driving their cancer and to get genomic profiling of their tumor. We want to know where these patients are and to meet them and their caregivers wherever they are in their treatment journey. And then to be able to offer them a precision medicine like cerebrentumab to tackle that cancer. So in summary, I've just shown you that NRG1 fusion represents a rare but actionable alteration across all solid tumors, including cholangiocarcinoma. Unfortunately, tumors that harbor NRG1 fusions don't do so well with the standard of care chemotherapy that we have nowadays. And so we really need to do better in order to develop new medicines to treat patients. NRG1 fusions can be detected, but it's best detected using an RNA-based sequencing where we can capture lots of genes at the same time versus a panel where we can look at 1 or 2 or up to 7 genes, but looking at the entire 22,000 gene at once. We know that NRG1 fusion overactivate HER3, and that sends a signal to the cancer cell to grow and divide. So if we can block HER3, we can potentially shut down that cancer growth and proliferation. And that's what we set out to do with the Creststone phase 2 study with cerebrentumab in adult patients who harbor NRG1 fusions. And we have clinical trial sites all over the country, as well as in Canada and Australia. So if you'd like to hear more about learn more about Creststone, you can go to nrg1fusion.com. We have a page that's also friendly for patients and caregivers, as well as physicians. And then of course, our team is available to answer any question that you might have, either about your cancer in terms of genomic profiling, and we can advise you in terms of how to talk to your doctor, how to get genomic profiling, or if you have your tumor profiled and had NRG1 fusion identified on it, we would love to be able to help you find a clinical trial site and potentially offer you Cerebentoma if that is appropriate based on discussion with your oncologist. So I'll stop there and see if we have any questions, but really, we're so excited to be partnering with the Cholangiocarcinoma Foundation. I know my team will be at the annual conference in person, as well as having an online presence. So we look forward to meeting some of you through that conference.
Awesome, that was great. Thank you so much. And that's exciting that you can open up a trial closer to the patient. I love to hear that. Okay, so we have a couple questions. Do they see NRG1 fusion often in folks that express KRAS?
Yeah, great question. And so, if NRG1 fusion is found, it's typically the one and only thing that's driving that cancer. So most tumors will be KRAS wild type. Just as in pancreatic cancer, most tumors, pancreatic cancer, express KRAS mutation. So only a subset harbor KRAS wild type. And it's like NRG1 fusions are more enriched in those KRAS wild type, but certainly it's not impossible. So. Okay.
So I think this question is one that I kind of had too. What about patients with mutated or overexpressed HER3? Is it Cerebentoma be appropriate for them? Was that the right drug?
Yeah, that's a great question. So, Merrimack, the previous sponsor, did look at various tumors that overexpress HER-regulin or NRG1, as well as HER3. And it didn't seem, because remember I said that amplification or gene overexpression, if they're common, the prevalence is high, but the likelihood of targeting that is lower because it might not be the driver. And so really with the monotherapy approach and the antibody approach with Cerebentoma, we think that it's really going to benefit those tumors that have NRG1 fusions. Now, it's possible that, we could evaluate this in patients with HER3 mutations and another approach or in combination, but for Cerebentoma as a monoclonal antibody, it seemed to be more beneficial for tumors that harbor NRG1 fusion over expressions of other genes, or HER3 or HER-regulin.
Okay, so I'm pretty sure you just answered this question, but I'm going to ask it anyway with what you just said. So you can have a HER3 or a HER2, but not necessarily the NRG1 fusion. And if you have the NRG1 fusion, you most likely will have the HER3 expression. Is that correct?
Yeah. So HER1, HER2, HER3, HER4 are normally expressed on cells, and in some cancers, those ERB family are overexpressed, or they can be mutated as well, and that leads to uncontrolled growth and proliferation. And then neuregulin or NRG1 is the protein that activates HER3 so that it would dimerize or partner with HER2 and sends the signal.
Okay. So you mentioned a preferred method of testing for NRG1. Is that form of testing different than what is traditionally done by Foundation One? And that was going to be my question too. So people who haven't had the RNA sequencing done, is that something they should go ask for to be tested again?
Excellent question. So comprehensive tumor profiling would look at both the DNA as well as the RNA of the tumor. And so to the best of our knowledge right now, Foundation Medicine uses a DNA-based assay. And so it can miss up to about 50 to 60% of fusions, not only NRG1 fusions, but other fusions as well. Other companies like Caris and Tempest, they will do both DNA and RNA based sequencing, or sometimes they'll look at the DNA and there's nothing found, then they would reflex to RNA. But we have learned that, for fusions, whether it's NRG1, FGFR fusion, RNA-based NGS is the best method to detect these fusions.
Okay, thank you. And I think you said roughly about 1% of cholangiocarcinoma patients have the NRG1 fusion. Is that correct? Is that what?
Yeah, it's a little less than 1% if you combine both cholangio and gallbladder, biliary tract, all the biliary tract cancers together. It's about less than 1% of those cancers.
Okay. Would patients who have already received Y90 TheraSpheres be a candidate for this study?
So in terms of our eligibility, we do not have an exclusion in terms of liver targeted therapy with Y90.
Okay, so yes, they would be eligible.
Yes.
Can it be detected in a circulating tumor DNA and tissue?
Great question. So liquid biopsies, that technology is evolving and continues to evolve. So we know that we can use liquid biopsy to detect some fusion. In terms of NRG1 fusion, I'm not aware of any panel right now that have NRG1 fusions detected in liquid biopsy, but that's something that we would certainly be interested in and hopefully support the research on that, so that, of course, liquid biopsy would be much better for patients and also for us to be able to follow the fusions throughout treatment.
Okay. I invite anyone to put any more questions out there that you might have. I think all the ones I wrote down have already been asked. And how do you suggest we go back to our physicians and ask for an RNA test if we haven't had one?
That's a great question. And so, I think, and as speaking as an oncologist myself, seeing patients, and as well as, having a loved one go through diagnosis of cancer and being an advocate for yourself, for your loved ones. And it's really, any doctor should not be offended. We do have some resources on our website, nrg1fusion.com. But I would just ask, has my tumor been sequenced? If not, is there an opportunity for us to get genomic profiling of my tumor so that we can know what's driving it and potentially have access to a clinical trial or to a medicine that target that particular alteration that is found? It does require, right now for NRG1 fusion, it does require tissue biopsy. But that doesn't mean that if you have tissue already that was taken at time of surgery or another biopsy, that might not be sufficient to send for sequencing. So I think, open dialogue with your physician. And like I said, any physician should not be offended that their patients advocating for that or asking about that.
Okay, and so as part of this study, are you doing the RNA assay test as part of the clinical trial, or do patients need to have that already completed?
Yeah, so we were rolling on on the local testing because, again, we're not doing the central confirmation proactively because we know patients need to start treatment right away. So our patients have the tests that identified NRG1 fusion. And then we enroll patients and start treating patients based on that test. So you haven't had genomic profiling, that's where talking to your doctor. If you've had previous genomic profiling, but it didn't reveal any alteration, whether that's NRG1 or any other fusions, you might ask, was this comprehensive and was this RNA based? Because RNA-based sequencing is the gold standard to identify fusions that may be present.
Okay, thank you. If one has the KRAS mutation, how would you know if it is the wild type and thus potentially have the NRG1 fusion, if she understood that correctly?
Yeah, great question. And so again, it's through sequencing of the tumor. So you can either do sequencing just for the KRAS gene, and that will tell you if there's a mutation or it's the wild type, or through these panels, comprehensive genomic profiling, it will tell you about the KRAS gene and all other genes that are in the tumor as well. And so really that's why we advocate for comprehensive genomic profiling versus over testing one individual gene or a panel of 7 to 8 genes. Because again, tissue is the issue. And so, right now with liquid biopsy, we're not quite there with the technology. So we do need the tumor for sequencing. And so patients' tissues should be used, appropriately. And that's where a conversation with your physician in terms of, has my tumor been sequenced? Is there an opportunity to get my tumor profile in case there's an alteration that's present that could be targeted by one of these agents that's in clinical trials?
Okay, this is a good one. Similar to FGFR2 fusions, can this treatment cause or create resistant tolerance mutations to energy 1?
So is the question about FGFR fusion or about?
Similar to the FGFR2 fusions, can this treatment cause or create resistance tolerance? So yes, about energy 1, can you get resistance with energy 1?
Yeah, again, another great question. So we believe that all tumors will develop resistance. It's just a matter of, are they resistant from the beginning or do they acquire resistant? And that's why, we need more treatments and different treatments to target the cancer. And so thus far, we don't have, in terms of what are the different mechanisms of resistance to Cerebentoma in NRG1 fusion positive tumors. We're still learning about that as we treat patients. There are studies, of course, at large academic centers that are looking at scientists in the lab, as well as oncologists in the clinic in terms of treating patients with HER3 antibodies. So.
Okay. If you have a driver already, is there a reason to seek further testing?
Great question. So based on what we know in the literature, currently, our knowledge is that if NRG1 fusions are mutually exclusive of other driver alteration, so it's unlikely, but there could be tumors that then develop NRG1 fusion as a mechanism of resistance to other driver. For example, there was a paper that says NRG1 fusion is a mechanism of resistance to ALK fusion inhibitors. So I would say that, typically NRG1 fusions are mutually exclusive of other oncogenic drivers, but there are tumors out there that may acquire NRG1 fusion as a mechanism of resistance.
Okay. So thanks to info on the foundation website, they requested genomic testing just prior to the biopsy and only last week received the Tempus results. The tumor tissue was tested, no normal tissue sample was submitted. Would additional tests on normal tissue be necessary to participate in this study?
No. So we don't need information about the normal tissue. Is really the somatic alteration or the tumor's genomic profiling. Some of those laboratories also want patients or sites to send in normal tissues just so that they can look at germline versus somatic, which belongs to the tumor. So our study will enroll patients with an NGS report that reports out NRG1 fusion in the tumor.
Okay. Is there a specific name to the clinical trial we can use to let our local oncologists know what to research?
Great question. So if Creststone, if they type in Creststone or Cerebentoma clinical trial, and likewise, you can go to nrg1fusion.com. And then I have the clinical trial number listed at the bottom of the slide as well.
Yes, and I'll get that information from you as well. And if anyone has questions, don't hesitate to reach out to us and we'll help you get connected. I think that is it for questions. If anyone else has a last question, throw it in the chat, but I think we've answered all of them. Great questions as usual. Thank you, Dr. Jansen, for presenting this and letting us know about this clinical trial and this opportunity. And yes, we want everyone to have their genomic testing done. And now we know to ask for RNA sequencing so we get all of the results that are important.
Great. Thank you so much for having us. And we're really excited to partner with the Cholangiocarcinoma Foundations, patients, caregivers, oncologists, and really, every patient matters.
Yes, thank you. And we look forward to seeing your team at the conference in a couple of weeks. You guys take care and have a great rest of your week.
Great. Thank you.
Thank you.